Hi, and welcome to our lecture on the process of development. We're going to take a look here at the process that occurs between conception and birth. However, due to a lack of time in a course like this one, we're going to be primarily focusing on some of the earlier stages. We're actually going to start even prior to conception or fertilization with the process we refer to as capacitation. And capacitation is going to be the getting ready of the sperm for the fertilization process. This process of capacitation takes place within the female reproductive tract and involves things like removal of the outer cholesterol coating and basically this preparation for the release of this acrosomal cap we see down here, which will happen as this sperm actually burrows into the egg that it's going to fertilize. Now, of course, we do release millions of sperm and only one of them fertilizes the egg, but they do have to make their pathway there. The reason why this might be important is if we're going to do any kind of artificial insemination, we're going to need to make sure that prior to that fertilization process, we undergo some form of capacitation or washing of the sperm to make them ready for fertilization. Our next topic is actually a little bit of a confusing one. We want to think about the boundary events for human gestation. And we commonly think of gestation and pregnancy as being synonyms. So we're really saying, when does pregnancy start? When does pregnancy end? And this is typically defined as the human gestation period from conception to parturition. Or we can say this again as fertilization to birth. Now, you might be saying, well, what's so confusing about this? And there's actually a couple of things. Number one, we don't know when conception takes place. And number two, this idea of conception and even some of the early stages of development don't actually produce the indication of pregnancy or even often result in pregnancy. So we actually kind of have a definition for the timing of pregnancy which doesn't actually take into account when pregnancy literally occurs as a result of changes to the body. So let's take a little bit more of a look at this and figure out why this might be confusing. So the average human gestation measured from this kind of event, from conception, is going to be 38 weeks. Now, this is kind of one thing that's interesting about this. We can potentially, if we know when conception occurs, start to talk about this 38-week process and consider the developmental process in light of this. In fact, we kind of have to, because if we're going to track the actual development of this product of conception, it will begin at the conception point rather than where we actually often start tracking this from. So the beginning event for a 40-week gestational calendar, which we commonly consider, is the start of the last menstrual period of the woman who is now pregnant. And there's kind of a reason for this if we think about it. We don't know when conception took place, but menstrual periods are something that we can track. And if we take a look at this, we can see that as we follow hormone levels, we can take a look here at the ovulation occurring at about day 14. Now, we can think about this as far as the menstrual cycle as well, that whether we're looking at the pituitary gland being affected here by things that are going to be released by the hypothalamus, and then the pituitary then affecting the ovary, or if we're looking directly at the ovary here, we're going to see the same thing at day 14. So we're going to have a spike in luteinizing hormone just prior to that, which we might pick up in an ovulation test, and then ovulation will end up taking place, and soon after ovulation is when fertilization will take place. So this happens roughly 2 weeks after the start of the last menstrual period, and that's why a physician would, of course, ask a mother when her last menstrual period started. That would give them an estimate of when this fertilization or conception process took place. Now, it ends up that we do often break up our pregnancy into trimesters. We're not going to spend a great deal of time on this, but we do want to recognize that we do have a kind of risky period in what we call the first trimester. And mothers, of course, are probably familiar with this. Often there's a delay between knowing that you're pregnant and informing other people that you're pregnant, because most miscarriages will take place during this first trimester. And we'll actually be able to figure out why that is as we take a look at the developmental process. But we do get the idea that the first trimester is kind of going to result in not that much growth of the uterus, but there is going to be significant development from the product of conception to something that by the end of this would actually look somewhat like the later products. You can kind of get the idea that the second couple of trimesters, while there will still be some developmental milestones in them, will often be dominated by a large amount of growth. And we see significantly less growth in that first trimester, and there are some good reasons why this would be the case. So when we think about this as far as breaking up developmental stages into kind of a set of 3 stages, or stages that really mark what's going on with development, we do this in a little bit different way than the trimesters. We break them up into pre-embryonic, embryonic, and fetal, which actually gives us some terms to refer to the product of conception as. So in the pre-embryonic stage, we don't yet have an embryo. It's going to be during roughly the first 2 weeks after fertilization. And of course, this is going to be largely focused on the initial cell division and some following up dividing of the cells, and ending kind of in this process of implantation. Now, in between those 2 periods is going to be something we call embryogenesis. And of course, this basically means that we need to form the embryo in order to be able to move on to the next stage, or the embryonic stage. So we'll kind of follow this up in a little bit more detail, but you can kind of get the idea here that we go from having this egg here to getting this fertilization process. As this fertilization process works, we're going to have the joining of nuclei from the egg and the sperm, resulting in a single cell we refer to as a zygote. The cleavage process will begin after that, which is going to be a division process, resulting in 2, 4, 8, and then 16 cells. That will be the morula, which is a solid ball of cells, and each cell is essentially the same. This is the product which will kind of arise at the uterus. So you can see all these earlier things are happening within the uterine tubes. And this morula then will hollow out, and it will actually develop 2 different kind of cell lines, a group around the outside, and then a cluster somewhere within one side of this. That's going to be where we actually have, for the first time, cells that are going to be destined to become the embryo, what we would call the embryoblast here, and cells along the outside referred to as the trophoblast, which will be destined to become this kind of component of the placenta that is developed by the fetus. You can see that as we kind of emerge from this, we start to get this embryo-looking structure here, but in no way have we made any kind of progress towards looking like a human at this point, but rather we would look pretty much like any other kind of developing vertebrate embryo at this point. The second stage, the embryonic stage here, is going to range all the way from something that in no way looks human or even necessarily like a mammal, to developing these kind of basic vertebrate characteristics, ultimately starting to look more mammalian, and eventually looking human by the end of this process. So this is kind of a set of embryos referred to as the Carnegie stages, and they do this to kind of track this developmental process. We can see kind of some of the things that occur along here, such as the presence of a post-anal tail typical of our phylum Chordata here. You can see the hind and front limb buds, but the tail will eventually disappear. We can see that there's very little of that left by the time we get through this. We also see kind of the loss of these little pouches within the pharyngeal region. So we have some kind of early traits we would share in common with a lot of animals, but we do actually emerge from this embryonic stage, and of course we'll be stepping into the fetal stage, looking somewhat like a fetus. So this stage again is from week 3 at the end of the pre-embryonic stage until the end of week 8, meaning obviously that at the beginning of week 9 we will begin that fetal stage. And the key reason why this is when most miscarriages occur is what's happening in here. The body is going through organogenesis, so it's going to be developing basically all of the organs that are going to be developed, and it's going to get that start of it. And it's going to start from a relatively simple state where it simply exists of embryonic tissue that is different than that which is going to become the placenta. So this little diagram off to the left shows some of the other things that have to happen in here. There's going to be folding in a couple of different directions. We're going to see that we have this caudal and cephalic folding where the embryo has to kind of curve around and it starts developing some of its shape, but it also has to actually kind of create things like a neural tube. It has to create its gut and fold over to actually create what we refer to as the coelom, the hollow body chamber inside of us. So you start from a flat disc and you have to create all of the three-dimensional shape that we know as being part of who we are within this embryonic stage. Now the fetal stage then is going to begin at week 9 and of course continue until birth, and it is going to continue some processes of developing organs, whether that be eventual ossification or kind of refinement of some of the organs that have been developed. Certainly towards the end of this, we're going to get significant brain growth. But those organs are basically all in place by the time we get here. So we can really say that this is largely about growth and then, of course, the increasing function of some of those organs. And again, even as we begin this process right here at the very end of the embryonic stage, or that boundary between embryonic and fetal, we are really looking relatively human. We can see digits and everything like that developed in here. And of course, we just become more and more human looking through that fetal stage. So let's go ahead and pick up in that pre-embryonic stage and find out why we wouldn't necessarily say that something like a miscarriage would take place at this point, but rather that we are getting a developmental process that is largely free of what's happening in the female. And that, of course, is because as we go through this fertilization process and result in this zygote, we're in the uterine tubes, generally here near the ampulla of the uterine tubes, so we're not actually attached to the mother yet. That means that chemicals that the mother might be taking in from smoking, drinking, whatever else, are not likely to have an effect on this portion of the developmental process. That doesn't mean that nothing can affect this. It just means that by not being attached, we're not getting those things directly from the mother, as will happen after implantation. So the zygote is this single-celled stage. Our next kind of really significant stage following up on the process of cleavage is the morula. This is what's going to arrive at the uterus. It is a solid ball of cells. And of course, we have no cell differentiation at this point. So the morula is cells that can become anything. And by becoming anything, I don't just mean anything in the human body, but can also become the placenta. So this is kind of a significant stage here, because as we jump into our next stage, the blastocyst, we do get that differentiation. So this morula will take on fluid, allowing this to hollow out. It's going to then produce two different cell lineages. We have those that become the placenta, referred to at this point as the trophoblast, and those that will become the embryo, referred to at this point as the embryoblast. And again, as a reminder, the trophoblast is going to be on the outside here, which makes sense since it's going to burrow into the endometrial lining of the uterus, and the inner layer of cells, which starts to thicken, is the embryoblast. Now, the kind of last stage we're going to be taking a look at here is actually a boundary between the pre-embryonic and embryonic stages. Of course, at the blastocyst stage, we have developed the embryoblast, but it's really not very, I guess you'd say, differentiated. It doesn't have much going on for it. It's all kind of one group of pre-embryonic cells. And gastrulation is going to be kind of a significant stage that bumps up the complexity and prepares for the future developmental processes. So one more thing we want to say about the blastocyst before we move on. Generally in the United States, those cells that are frozen for in vitro fertilization clinics are at the blastula stage or the blastocyst stage. So that particular stage is going to be what we freeze, and again, the key thing here is it does have embryonic cells, and those are ready to be implanted in a uterus because gastrulation, the process of forming a gastrula, takes place during the implantation process. What we can see here from this is that we actually have above this yolk sac here, this layer down below, and this is going to be a layer of what we're later going to call endoderm. And that endoderm then is going to have on its opposite side here a group of cells we refer to as the ectoderm. And they are going to have some migration of cells in between those two layers to form what we call the mesoderm, or the middle layer. Now we can actually follow these particular cell layers, these what we call 3 germ layers that are produced during gastrulation, throughout the developmental process and figure out that they become different kinds of tissues. So during that organogenesis process of that embryonic stage, we're going to have the ectoderm give rise to the epidermis, which makes a lot of sense. Ecto means outside, epidermis is the outside layer, but also some of this is going to burrow in and pinch off and become the nervous system. So the ectoderm gives rise to both nervous system and the epidermis. The mesoderm is primarily going to become things like the dermis, the skeleton, muscle, things like bone marrow as well, giving rise to lots of blood vessels, portions of some other organs, as well as our mesothelium. So we can largely think about this as becoming connective tissue. Now, this is a little bit confusing because obviously many of these organs are also going to have some inner linings, which do not always arise from this mesoderm. So we can kind of see that here, and again, I would probably focus mostly on the muscle and the bone, but recognize that that is in no way the limit of what we're getting from this layer. The endoderm then is going to become largely things like the mucosal epithelia, the linings of our digestive, respiratory, and reproductive tracts, and of course a few other things that are associated with some of those, like some of the glands that are associated with them. So the endoderm is kind of that inside layer, in this case, the lining of some of these very significant organs. Now you might be saying, why does it really matter if we kind of learn what these different layers give rise to? But if we think about the process of potentially forming organs for transplant, if we're trying to be able to actually produce these kind of things, if we want to be able to do that from stem cells, such as those cells that we might find in the blastocyst, well, we would actually need to be able to drive those stem cells, which could become anything in the body, through different stages. If we want to form the lining of a respiratory tract, we would need to take those stem cells and have them differentiate into endoderm, and then have them differentiate into the particular lining cells that we wish. So we actually can follow how all these changes work, and that may help guide us, and we could say actually that it is helping guide us to the formation of organs in this manner. Now there's another topic that we want to address at this time, and that would be the extraembryonic membranes. These are things that are going to surround the membrane. These are things that are shared amongst all the amniota. That would be all the land vertebrates other than amphibians who have an amniotic egg. So whether we're talking about an egg like a chicken egg, or in a mammal, this kind of retained egg that then develops within the mother, we're going to have a set of membranes. We're going to have things like a yolk sac, although ours will not contain yolk because we're not growing off of that. We would have this wrapper around the embryo, which we call the amniotic membrane, or the amnion, and then we would have this chorion here, which is largely involved in some exchange of things like gases and nutrients. So let's take a look at these one by one, starting here with the amnion, the one that people should probably be most familiar with. It's the one that contains the amniotic fluid, that fluid that surrounds the developing embryo. So we've got our embryo inside of here and this fluid-filled sac around this. So this will actually break when we say that a mother's water breaks and release that fluid there, generally not very long prior to the birth process. Often the water breaking will be initiated by the beginnings of labor. Of course, this allows for movement in here and protection and a kind of nice constant environment for the embryo to develop within. The yolk sac is not the most important one, let's say, in humans. We also have something called the allantois, which you can't really even see very well in us. The yolk sac, of course, doesn't contain yolk. It's not our source of nutrition. That's going to eventually be the placenta. But the first blood cells are formed within here. And so it's really kind of a significant component that we keep. And it's just interesting that we can see this kind of piece of our evolutionary heritage, even though we don't actually use the yolk for nutrition. Now, I would say that the chorion, like the amnion, would be one of the really significant ones, and in mammals, perhaps maybe the most significant. So the chorion is going to produce villi, these things that have all this additional surface area, and it's going to burrow into the endometrium of the uterus. So we can see these extensions of this chorion here, each one referred to as a chorionic villus. We've got the uterus over here with its blood vessels, including its spiral arteries, and we're going to bring these pools of maternal blood that these chorionic villi sit inside of. And that's going to allow exchange of gases, nutrients, and wastes between the maternal blood and this developing embryonic or fetal blood without the direct exchange of blood. So it's a really, really significant feature, and of course for placental mammals like ourselves, it is one of our defining characteristics. And so this is developed by the chorion, which was originally the trophoblast. And that takes us to this idea of a placenta. And we can see this up a little bit more closely. Of course, we remember learning about the umbilical vein, which returns from the placenta, and the two umbilical arteries, which provide blood to the placenta. This is another example, like in the pulmonary circuit, where we had the blue vessels, the ones that are low in oxygen, being arteries, and the one that is high in oxygen being a vein. But we basically take this fetal blood to these villi, and we're going to have that exchange here. Of course, you can see maternal veins and maternal arteries, so we bring in this kind of nice, fresh arterial systemic circuit blood from the mother into these pools. We get exchange across there, then we drain that blood back out to the mother. And of course, returning in red to the fetus in that umbilical vein will be that high oxygen, high nutrient blood that will feed the developing embryo and fetus. Now, it's a little bit weird to talk about this as an embryo and a fetus being kind of fed by this placenta, and it is actually a little bit complicated. So after roughly week 8 or so, which is going to mean that we're talking as we enter that fetal stage, the placenta is going to take over as kind of the primary source of nutrition. We actually derive some nutrition directly from the trophoblast, which would be that chorion, or those chorionic cells around the outside of that blastocyst at first, and then the gastrula. But that trophoblastic nutrition, the directly deriving that from the cells of the uterus, is going to be relatively short-lived, and we decrease the significance of that as we see the placenta grow. But as you can see here, the placenta is going to continue to grow and be able to provide more nutrition as the fetus develops and, of course, gets larger and requires more nutrients. Therefore, it would be difficult to say something like, well, the placenta is developed after 8 weeks. Well, it takes over as the primary source of nutrition then, but it continues its developmental process as the fetus continues its. And here we can kind of just get a look at the placenta. So here we have a kind of developing placenta. We've got some chorion on the outside. This is just a nice view of the amniotic sac surrounding this developing boundary organism between that embryonic and fetal stage. Here we see the placenta with the kind of rough, fibrous side towards the fetus and the cord coming off of that. And here we see the uterine side, which looks a little bit more, I was going to say kind of fluffy, or like it's a little bit softer. And that's, of course, because of all of those villi that are increasing the surface area there, and the fact that this was actually embedded in the endometrium of the uterus.